I had the Christian worldview portion of this assignment. I chose to look at Sirach 38, verses 1 through 4. Honor physicians for their services, for the Lord created them, for their gift of healing comes from the Most High. They are rewarded by the King, and the skill of the physicians makes them distinguished, and in the presence of the great they are admired. For the Lord created medicine out of the earth, and the sensible will not despise them. I think this shows how the use of antiviral drugs and antifungal drugs is encouraged because they are made from the earth, and the earth is God's creation. So these drugs are a part of the creation and made from the earth and God's creation. So I think it's encouraging people to use these drugs to help with their viral infections and antifungal infections to help treat them, and so they get better. Another verse I found that helps support this is Proverbs 17, verse 22. A merry heart does good, like medicine, but a broken spirit dries the bones. I think this quote is showing how medicine is good and encourages people to take medicine, because the Bible encourages a lot of merry and forgiveness to others, and I believe that by comparing medicine to a merry heart, it shows how good medicine is and how people should take it to help cure themselves and make them better.
Viruses are obligate intracellular parasites. They rely on host cells to survive and replicate. Viruses don't have a cell wall and they lack ribosomes. Viruses are completely unaffected by antibiotics. Antibiotics work by attacking structures or processes in bacteria like cell walls. Since viruses lack these structures, antibiotics cannot work on them. Antivirals are only effective against replicating viruses and a specific type of virus. Antivirals are designed to target specific viral proteins or processes unique to a particular virus. Antivirals usually interfere with specific steps in the replication processes. Viruses such as HIV and herpes can remain latent in cells. During latency, there's no replication machinery for the antiviral to target, so the medications for treating viruses do not cure them. Combination therapy involves the use of 2 or more medications to treat diseases that evolve rapidly, like HIV and hepatitis C. Combination therapy reduces the chance of the virus developing resistance and is more effective than a single medication. Entry inhibitors are a class of drugs used in antiviral treatment. Medications effective against HIV are entry inhibitors, meaning they prevent the viruses from entering host cells by preventing attachment or fusion with the host cell membrane. Entry inhibitors can be separated into 4 categories. Attachment inhibitor, which blocks the virus from attaching to the host cell and prevents the binding of the virus to receptors on the cell surface. Post-attachment inhibitor, which stops the virus after it has attached but before it enters the cell, and interferes with the process needed for the virus to penetrate the cell membrane after the attachment. CCR5 antagonist, which blocks the CCR5 receptor on the host cell, prevents the virus from using this co-receptor to enter the cell. Fusion inhibitor, which prevents the viral membrane and cell membrane from fusing, inhibits the virus's ability to merge with the cell membrane, blocking entry into the host cell. Viral encoding is a process that occurs in viral replication. There's 2 groups of medications that target the encoding process that releases nucleic acid of a viral particle from the protein coat. Medications target the encoding process because viruses cannot replicate and produce new viral particles without releasing their DNA or RNA. Capsid inhibitors. Capsid inhibitors work by binding to the protein that makes up the subunits that compose the viral capsid. This binding interferes with both capsid disassembly during viral encoding and capsid assembly during viral maturation, targeting the virus during 2 separate stages of its replication cycle. By interfering with capsid stability, they inhibit the virus's ability to replicate and infect new cells. This is useful for treating patients with multi-drug resistant HIV infections.
Many of the most effective antiviral medications focus on targeting viral enzymes essential for replicating the virus's nucleic acid. These medications include nucleoside and nucleotide analogs, non-nucleoside polymerase inhibitors, non-nucleoside reverse transcriptase inhibitors, N5-5A inhibitors, cap-dependent endonuclease inhibitors. Nucleosides targets are HIV and HBV, reverse transcriptase. Non-nucleoside polymerase inhibitors bind to non-nucleotide sites and are HIV specific. NS5A inhibitors target NS5A proteins. Cap-dependent endonuclease inhibitors target is viral mRNA through a cap snatching process. Another type of antiviral drugs are integrase strand transfer inhibitors, also known as INSTIs. INSTIs block the HIV enzyme integrase, stopping the virus from inserting its DNA into the host cell's genome. Some antiviral medications target viral enzymes needed to assemble and release new virus particles. Types include protease inhibitors and neuraminidase inhibitors. Protease inhibitors target is viral proteases required for polyprotein cleavage. Neuraminidase inhibitors
The target of most antifungal medications is ergosterol, a sterol found in the cell membranes of fungi and some protists. Ergosterol serves to maintain the integrity and fluidity of fungal cell membranes. Azoles, which are a class of compounds that function by preventing the synthesis of ergosterol. This leads to defective fungal membranes and leak cytoplasmic contents. There's 3 types: imidazoles, triazoles, and tetrazoles. Paulines, a group of antibiotics that are produced by certain species of streptomyces. Paulines bind to ergosterol, which disrupts the fungal membrane and kills the cell by leaking its contents. There's 3 types: allylamines, tolnaftates, and butenofine. These inhibit the enzyme in the pathway of the synthesis of ergosterol. These can be applied to the skin to treat infections, but some can be taken orally.
All right, so mechanisms of antifungal drugs. Antifungal drugs will actually disrupt the cell synthesis or the cell wall synthesis by inhibiting certain enzymes. When these enzymes are inhibited or stopped or prevented, it'll actually impact the cell wall by not allowing the fungus to build or maintain the cell wall, and because of this, it becomes unstable, and this can result in lysis or cell death. Later on, cell death. Um, the introkindins or caspofungin is one of the components that will inhibit the enzymes that is responsible for the sensitization of beta-1,3 glycan. And then also you have sarcopyrox, which interferes with the stability of the membrane of the fungal cell. And then you have other importing, inhibiting vital components of the fungal cell wall is also going to be beta-glucan and of course beta-1,3 D-glucan synthase. It also disrupts the microtubules by utilizing glycopholin, which binds to polymerized fungal microtubules. This prevents depolarization, and this results depolymerizing, and this results in the fungal cell becoming unstable. And because it becomes unstable, the fungal cell actually wouldn't be able to replicate. And then other examples of these drugs are capsulfungin, mycofungin, and andifungin. All right, so mechanisms of antifungal drugs. Antifungal drugs will actually disrupt the cell synthesis or the cell wall synthesis by inhibiting certain enzymes. When these enzymes are inhibited or stopped or prevented, it'll actually impact the cell wall by not allowing the fungus to build or maintain the cell wall, and because of this, it becomes unstable, and this can result in lysis or cell death. Later on, cell death. Um, the introkindins or caspofungin is one of the components that will inhibit the enzymes that is responsible for the sensitization of beta-1,3 glycan. And then also you have sarcopyrox, which interferes with the stability of the membrane of the fungal cell. And then you have other importing, inhibiting vital components of the fungal cell wall is also going to be beta-glucan and of course beta-1,3-D-glucan synthase. It also disrupts the microtubules by utilizing glycopholin, which binds to polymerized fungal microtubules. This prevents depolarization, and this results, depolymerizing, and this results in the fungal cell becoming unstable. And because it becomes unstable, the fungal cell actually wouldn't be able to replicate. And then other examples of these drugs are capsule fungin, mycofungin, and andifungin.